there is third generation of EGFR tyrosine kinase inhibitors that have a very unique feature, which is the fact that they really specifically shut down the signaling from EGFR mutant receptors and not the signaling from EGFR wild-type receptors. So the EGFR wild-type sparing characteristics of these products make that they lack the classical skin and gastrointestinal toxicity while keeping the clinical activity in the EGFR mutant pathway. On top of that feature, for EGFR mutant receptors, not only they block the sensitizing receptors, but they also block the acquired resistant T790M conformational receptor change, allowing therefore to overcome acquired resistance to first generation and second generation EGFR tyrosine kinase inhibitors such as gefitinib, erlotinib, or fafatinib. So they really represent a game changer in the field of non-small cell lung cancer. There are two very advanced EGFR tyrosine kinase inhibitors of third generation, namely rociletinib, formerly known as CO1686 from the Clovis Pharma group, and AZD9291 from AstraZeneca. And then we have a more recent one that is going to be presented also tomorrow. The two most advanced compounds have indisputable activity in T790M positive EGFR mutant non-small cell lung cancer patients. If I focus on rociletinib, which is the compound I discussed during the TAT meeting, what I can tell you is that in T790M EGFR mutant patients, response rate is 67% with an estimated median PFS, which is still immature, of 10.4 months. So this is significant because to the 12 to 16 months of activity that gefitinib or erlotinib provide to an EGFR mutant naive patient, we're able now to add another extra year through the use of rociletinib, for instance. AZD9291 has similar activity. Now, the unique feature I did share during the TAT meeting regarding rociletinib is the fact that it also has activity in T790M negative patients, which is kind of surprising because you expect mostly to be positive in T790M positive patients. And three potential explanations can be provided for the activity that is in EGFR mutant patients but without the acquired T790M resistant mutation. First, these might be false negative T790M. Second, these may represent tumor heterogeneity. Or third, this is related to the very unique mechanism of action of rociletinib, which not only has the EGFR inhibitory properties, but also has a metabolite, M504, who has intrinsic IGF-1R inhibitory capacities that could provide this specific advantage in T790M negative patients where objective response rate was seen in 43% of them with a median PFS above 6 months. That's the $1 million question is, are these compounds going to be used after failure to erlotinib, gefitinib, second line or beyond? That's the faster way for registration of these compounds. But both companies, AstraZeneca through the AURA program and rociletinib, Clovis through the TIGER program, are also randomizing their compound to first generation EGFR tyrosine kinase inhibitors in the frontline setting, but that is going to take some extra months to be addressed. On top of the activity both in T790M positive and negative patients, that trial had a very nice biomarker readout, which is based on the blood draws that were analyzing by beaming the presence of T790M mutations. And interestingly enough, we did find some patients who were T790M negative in their lung, but positive in their blood. As you know, TAT is a very unique meeting. It's a small size meeting, below 500 people, one single unique session, so there is no need to get lost in choosing A against B or against C. Very friendly atmosphere with easy interaction between key opinion leaders, attendees, academic people, and pharma industry people. I think this year we had a significant number of interesting fields that were addressed. Of course, immuno-oncology, a huge wave of excitement there, not only through PD-1, PD-L1, but also the combinations of CTLA-4s and PD-1s. Also, the new way to evaluate radiologically response to these compounds across tumor growth rate or pseudoprogression evaluation. On top of immuno-oncology, on top of the new radiological readouts, we also had a very nice session on cyclin-dependent kinase inhibitors that seemed to be a potential way to modulate super enhancers like the MYC amplification. And we also had a very, very promising session on epigenetic modulators, the bromodomain modulators, both EZH2 and a BRD4 modulator, both from Oncoethics and GSK, appeared as having indisputable activity and promising efficacy data. I invite everyone to attend the TAT meetings. They are probably a very easy meeting in which junior people can mingle with established investigators, in which established investigators can create very easily networks of interaction with pharma people. This is a meeting where, contrary to the very large meetings, has a very friendly and enjoyable atmosphere. And of course, coming to Paris is always a fantastic setting. We have a rule: we don't go over 500 people. So first come, first served. Very simple, very basic, very French. TAT 2016 will be done under the leadership of Professor Pepe Jacquard, with as host organizing institution, the Georgetown University. We're going to alternate TAT between Paris and Washington. So, you know, the capital of the US, what it has to offer, and I am sure fantastic science there too.